So in the third part of our lab, we would have done our inoculations onto a healthy tomato, and we would have been making observations over a 7 to 10 day period. So here we have our samples. This is our tomato that was inoculated with our unknown specimen, with our organism. So you can clearly see signs and symptoms of disease present. And here we have our control. Okay. So for this session, what we would have to do is observe our diseased tomato, look at the signs and symptoms. You will look at things like the lesions, the discoloration of tissue, and so on, and compare it to our first sample to compare and see whether or not the signs and symptoms were the same. Additionally, at a microbiological level, we are going to inoculate this sample onto a nutrient agar, allow it to incubate for 24 to 48 hours, and then we will be looking at the colony morphology. We'll carry a gram stain on that organism and look at colony morphology to compare that to our results from our previous colonies that we would have obtained from this organism. The idea is that the organism that would have caused the disease in our first initial sample is the same organism that we would have isolated from that lesion, placed and re-inoculated into a healthy organism. It caused disease in that healthy organism, and when we isolate it, it will be identified based on colony morphology and characteristics that it was the same organism causing disease in that first tomato. So again, we have our inoculating loop. We are using aseptic technique to take a sample of this diseased tomato and do a quadrat shriek, which we will be looking at in 24 to 48 hours for the growth of our pure cultures, doing a gram stain and looking at the morphology of that organism or the characteristics of that organism. So following with our first lab, same procedure where we are getting our sample and doing a quadrat shriek. We flame our loop until red hot. Put alcohol. Flame off our alcohol. So very carefully, now you can see we have a lot of disease going on in the sample. So we insert into the region where we inoculated. We perform a quadrat shriek. Flame our loop before we put it away, re-sterilize. And we label our sample. Alright, so we will label this Koch Postulates lab number 3. We put our date and our name on it. Now this lab was done before, so we see here that we do have a sample that was inoculated for 24 to 48 hours. We do see that we would have growth taking place. When we looked at the characteristics of the colony or the colony morphology, it was observed to be similar to that of our initial organism. A gram stain was done on this specimen. It was viewed under the microscope at a 40-time magnification, and that image will be provided to you so that you can see whether or not the characteristics of that organism were the same as our first organism that was isolated. So that's it for your Koch Postulates lab. For this lab, after you have completed all sessions and looked at all the 3 labs that we would have done, the 3 laboratory sessions that we would have carried out, you are to write up a full report on the entire procedure, talking about how each of the stages of Koch postulates were carried out to show the association of a disease with different signs and symptoms present on a living organism. Thank you.